So hi everyone, this is going to be part 2 of the sensory system series, specifically focused on the eye. I got some feedback after the previous video and it seems that everyone wanted me to focus on more difficult concepts of each section and just skip over the minutiae. So I totally agree with that and I hopefully will try to do that today. So let's get started. So there are going to be 3 topics we'll be focusing on today: the retina, the conversion of light, to information, and the processing of the visual world. So let's start with the retina. The retina will be split into 3 aspects. So the first part we'll be talking about is the appearance. So now when a physician or a nurse does a fundoscopic exam, what they're doing is looking at the back of the eye to see what's going on with the retina. And what they're looking for is problems with the vasculature in the retina, because that's basically how the pathophysiology manifests. And if you guys are interested, you should look into diabetic retinopathy. And in this disease state, what happens is the glucose molecules enters the vasculature of the eye and builds up, and eventually the blood vessels will rupture and damage the retina. And blindness is very common in this disease state, so it's important to note when you're thinking about this stuff. So what we're going to focus on are the cells present in the retina, and there's 2 types, 2 cell types. The first being the cones, and they're what allow us to see in color. And the next being the rods, and they're what allow us to see in black and white. So for this video, you're using your rods. Now, when I picture the retina, I like to picture a giant forest. Bear with me, it's a little weird, but so that's the retina. And I like to think of the cones as evergreens and the rods as, I guess, regular trees. So outside, near the edge of the retina, is what we call the peripheral retina. Now, the peripheral retina has a ton of regular trees, but very little evergreen. So as we get deeper into this retina forest, in an area called the macula, we start seeing more evergreen trees and less regular trees. And once we get to the center, in this place called the fovea, you see almost all evergreens and no regular trees. So as we get deeper, there's going to be an increase in the number of cones and a decrease in the number of rods. And off to the side a little bit, we're going to see a hole, and this is called the optic disc. And that's where all of the nerves from the cones and the rods exit to go to the brain. And this is also known as the blind spot. because technically the light that hits this area isn't recognized. So what the brain does is it mentally fills in what it would expect to be in the blind spot in your visual field. So the next part would be the cytoarchitecture. Cytoarchitecture of the retina. So with the cytoarchitecture of the retina, we'll be going from the deepest to the most superficial of the retina. So at the deepest, what we have is something called the pigmented epithelium. And what this pigmented epithelium is a modified layer of the choroid that absorbs any excess light that doesn't get processed. And you don't want this light to go to any special nerve cells in the brain, so it absorbs the light and it prevents any damage to the brain. Now, after the pigmented epithelium, you have the outer nuclear layer, which is comprised of the photoreceptor cells, aka the cones and the rods. And this is the outer nuclear layer. And after that, there's the outer plexiform, which is comprised of horizontal cells that amplify the signal sent by the photoreceptors. And after the outer plexiform, you have something called bipolar cells, because they're moody. Just kidding. They're bipolar cells because they have receptors at two ends, one facing the outer plexiform and one facing the inner plexiform, which contain amacrine cells that amplify the signal of the bipolar cells. Now, after the amacrine cells, you have this ganglion layer, which then send their axon out into the optic disc, and this one is called the ganglion layer. The ganglion layer contains the nerve cells which generate electrical signals, and that's called an action potential, while the rest of the cells down here communicate through chemical signals. Now, what's counterintuitive here is that light enters from the top and has to go through all of these cells to be recognized at the outer nuclear layer with the photoreceptors. The signal goes from the photoreceptors to the bipolar cells and then to the ganglion, which then sends the signals to the brain. To get into the circuitry, we have to understand that the retina is a duplex, meaning that it's comprised of two functioning aspects, which will be the scotopic and then the photopic. The scotopic retina activates in the dark and is comprised of rods. And these rods contain a pigment called rhodopsin. And that's a purple color. And then the photopic retina consists of cones and is activated during the light during the day. And these cones contain a pigment called opsin. And that's comprised of 3 different types of opsin, which are red, blue, blue, and green. And these three pigments is what allows us to see all the vibrant colors in the world. So looking at the circuitry, the rods will stimulate, one rod will stimulate multiple horizontal cells, and one horizontal cell will stimulate many bipolar cells, and one bipolar cell will stimulate many amacrine cells, and same thing, one amacrine cell will stimulate many ganglion, which then potentiates the signal to the brain. And this circuitry is called convergent. Convergent circuitry. As for the photopic retina, one cone will stimulate one bipolar cell, will stimulate one ganglion, and this circuitry is called one-to-one circuitry. And the reason for this difference in circuitry is because the scotopic retina is trying to get a visual image when there's very little light stimuli, while the photopic retina is trying to get a visual image when there's an overload of light stimuli. For the next concept, we're going to be talking about the conversion of light, or the photons reflecting off objects in our visual field into information that we can understand and process. And for this section, we'll be mainly focusing on the scotopic retina, or the activation of rods to help us see what's in front of us. A quote that really summarizes this concept and has stayed with me since taking Dr. Uzriak's course all those years back is, "The absence of information is information in itself." So that's a little bit of a difficult concept to understand. So I'll give you guys an example. Why are you guys going to class next week? Because there's no announcement for you to not go to class. So the absence of the announcement gives you the reason to show up for class. So with that in mind, let's look at what happens when a photon hits a receptor. So there's a photon, light, and it's going to hit the rod, and what you're going to get is a decrease in cyclic GMP, which is a secondary messenger. So what cyclic GMP does is cause a reaction in the cell. So in this case, since there's a decrease in cyclic GMP, there's going to be no reaction. Now in the absence of light, we get an increase in cyclic GMP, and this leads to sodium channels on the cell membrane to open, and sodium enters the cell, and this leads to calcium channels on the cell membrane to open, and calcium enters the cell, and with calcium it causes the release of a neurotransmitter, which is a form of chemical communication with another cell. So in our case, it's chemical communication with from the photoreceptors to the bipolar cells. What happens downstream in this process? So let's start with light hitting the rod. Hits the rod, and what you get is no neurotransmitter release. That'll signal two types of bipolar cells. There's on bipolar cells and there's off bipolar cells. So on bipolar cells, when there is no neurotransmitter, it causes the release of neurotransmitter. off bipolar cells, when there is no neurotransmitter, it won't release a neurotransmitter. So the neurotransmitter release from the on bipolar cells will always signal an M-type ganglion, and then the off bipolar cells will signal P-type ganglions. Now we can look at the same process when there's no light. It's going to work on the rods, and we know from before that in this case there will be neurotransmitter released, and that's going to signal on and off bipolar cells. And neurotransmitter on bipolar cells will not release neurotransmitter, and neurotransmitter release on an off bipolar cell will release a neurotransmitter. And on bipolar cells, once again, will be signaling to the M-type ganglion, and off bipolar cells will be signaling to the P-type ganglion. Now the axons of these ganglion will go to something called the lateral geniculate nucleus, the LGN, in the thalamus. Now there are two parts to the LGN in the thalamus. One's going to be the magnocellular layer, and then the other type is going to be the parvocellular layer. So the M-type ganglion will signal to the magnocellular layer, and the P-type will go to the parvocellular layer. The magnocellular layer is layers 1 and 2. The parvocellular layer is layers 3, 4, 5, and 6. The axons from the magnocellular layer and the parvocellular layer will then go through something called optic radiation, which is basically the pathway from the thalamus to the primary visual cortex. The cortex is comprised of 6 layers, and the axons of the magnocellular layer and the parvocellular layer will be sent to the 4th layer. Now the 4th layer is split up into 3 different parts: A, B, and C. And then this part C is split into 2 parts more: the alpha layer and the beta. So I color-coded alpha as maroon for the magnocellular layer, and beta as purple for the parvocellular layer, because the parvocellular layer's axons will then go to the primary visual cortex layer 4, section C, beta, and the magnocellular layer will go to layer 4, section C, alpha. And magnocellular neurons are in charge of movement, and parvocellular neurons are in charge of color. I'll finish up the rest for you guys in part 3, so hopefully I'll have that out by next week. Thank you.